Okay. Here are your three types of B cells. There's only one type of B cell. B cell is a B cell. This one we'll call it X, we'll call this one Y, we'll call this one Z. And the red thing here we have here is called the antigen. We designate antigen by capital A, lowercase g. It is normally part of the bacterial cell or whatever that caused the immune response. It is sometimes referred to the antigenic determinant, or the specific part is sometimes called the epitope. The antigen is what starts the immune process to occur. Now here's the definition I want you to know for what an antigen is. An antigen is anything which elicits an immune response. An antigen is anything which elicits an immune response. So could a bacterial cell elicit an immune response? Yes. Could a virus? Could a toxin from a snake bite? Yes. Could a bullet? Yeah, anything which can elicit an immune response is considered an antigen. That's how you define an antigen. So let's begin with this B cell. We have an antigen that comes in. And of these three B cells, these are all three naive B cells. They're just immature sitting there. Which of these will be activated by this specific antigen, X, Y, or Z?
Y.
Exactly, because Y has a fit. That fits. That demonstrates in the immune system the need or the result of specificity. Yeah, one B cell will respond to one specific antigen. So this antigen will not activate this B cell or this one, only this one. Now, once that B cell has been activated, it will start growing, maturing, and dividing into two types of cells. One thing it'll grow into is called memory B cells. These memory B cells are simply more of the same. Just build more of these. So the first time you get exposed to an infectious agent, antigenic agent, you may only have one B cell, for an example. But after that, you have hundreds or thousands of these same exact B cells because you've been exposed to this already. The next cell produced are plasma cells. Plasma cells function to produce antibodies. Designated capital A, lowercase b. There are 5 classes of antibodies. I'm not going to go over those in lecture. You can find those in your textbook. Does a very good job describing those 5 classes. These antibodies now float in the humerus, the lymphatic fluid, the lymph fluid, the blood supply. That's why this is called the humeral branch. Now these antibodies have several functions. One function is neutralization. Let's say here's your bacterial cell. Here's your antigenic determinant. One thing you learn in the summer microbiology is the first step required for bacterial infection to occur is adherence. The cell must adhere to your tissue. If it cannot adhere, you can't proliferate. So let's say in this case, the epitope, the antigenic determinant, is a site of adherence. Or, let's say this is the protein from a snakebite poison. The antibody would simply physically fit this, and now it has not killed the bacterial cell. What has it done to it? It's neutralized it. It can no longer cause any damage. If it's a poison, it'll neutralize the poison.
Does it destroy it? It just neutralizes it?
Doesn't destroy it, just neutralizes it.
Okay.
Exactly. Number 2. It causes agglutination. What happens here all these antibodies with the antigens attached start getting tangled up. They agglutinate. And next week we'll run an experiment on this on blood typing and share what agglutination actually looks at. But again, if they're tangled up, they cannot cause infection. They've been taken care of. Precipitation. They'll actually fall out of the solution. They get so large, they'll travel to the spleen. Well, they'll undergo immune clearance. The spleen will get rid of all of them. Antibodies and antigens, all the whole mess will be cleared out by the spleen. Activates complement. Did we go over the cascade of complement in this class Monday? Activates complement. Number 5.
Oh, yeah.
Opsonization. Which is what? Remember my apple bobbing?
The stem of the apple and the
Right. That puts a stem on the cell, so it makes it easier to phagocytize. So it increases phagocytosis. Couple things presented by this process. One is specificity. One B cell respond, or one specific type of B cell will respond to one specific type of antigen. Another is memory. Which is caused by the production of memory B cells. So the first time one has infection, let's say chickenpox, you'll get the disease, you'll have the symptoms, you'll run the disease. Because the first time you had an infection, you don't have quite enough antibodies to kill it off early. It kills it eventually, that's why you don't die of it, but you eventually conquer it. But why don't you get the chickenpox or the same disease the second time?
My daughter did.
Well, but we know you're strange. That's okay.
Because you build up.
And some people do. But generally speaking, why do you not get the disease the second time? You got the memory cells the second time. It's not it's not one B cell that becomes activated, it's thousands. So you have a very rapid response of antibodies, a very rapid response immune system. You'll get the disease the second time, you get the infection, but it's quelled or stopped very quickly. You stay asymptomatic. This is the function of vaccines. You get a shot. What the shot will have is simply part of the antigen or the antigen determinant. Your body builds build memory cells. So when you actually do get to the real disease, you have these memory cells, you stay asymptomatic. Okay, question about the B cell.
No, but so that doctor that's supposed to come in, I use that term loosely, doesn't like to have the immune shots. So he's thinking that we could, I mean, I just don't understand.
I don't know what he's thinking. I have no idea. I'll let you know when I hear it myself.
Are you going to go watch
Oh,
it?
yeah. Okay, one thing I haven't expressed yet, which is pretty important. The maturation and specialization of B cells into memory cells, plasma cells, normally will not occur fast enough to do any good unless you have chemicals released here, cytokines, from a helper T cell. We'll talk about T cells a little bit. This helper T cell releases chemicals which makes us start activating fast enough to be effective. Without the T cell, this is not happening fast enough in an efficient system. Let's see.
Now with AIDS or HIV, we don't develop the memory B cells? Or we
We'll talk
do?
about that. We'll talk about it when we get to T cells. The question is, how does HIV work? We'll address that in the T cell side just a second. One thing unique about this also, if you look, the B cells can be stimulated by direct contact with the antigen. That's not the case with T cells. T cells cannot be activated by direct contact, only with B cells. Okay, question before I go on to T cells. Okay, let's stop and do T cells.